I really advise the use of human-derived cells, like human organoids or human primary cultures, so that you can really take advantage of using the complexity of these cultures and the dynamic advantages of having this microfluidic system. Hi, I'm Nanci from Leuven in Belgium, and I'm a postdoc in the Porto Lab. And my research focuses on studying hepatitis E virus replication and dissemination in the gut-liver axis. And for that, we use organoids and organ-on-chip models. Hepatitis E virus is the main cause of viral hepatitis worldwide, and in contrary to hepatitis B and C, this virus is transmitted fecal-orally. So the gut is the first barrier that the virus encounters in our body to cause infection. But main of the research is focused on the liver because the main disease is liver hepatitis. But we see in chronic patients that after they stop therapeutics, the virus rebounds, so it's hidden somewhere. And the hypothesis is that the gut could be a reservoir for hepatitis E virus. So for that, we want to study how virus replicates and disseminates from the gut to the liver and see if there's a connection between these two organs that could be the cause for this rebound in untreated patients. So to study hepatitis E virus in the gut, we are using human intestinal enteroids. So these are adult-derived organoids cultures that we have studied in different setups. So in 3D, in their native conformation, in 2D in transval systems, or also by transecting these organoids by electroporation with the virus. And after, we also wanted to study the dissemination from the gut to other compartments, and for that we also established an organ-on-chip model. That here you can see the structure of this gut-on-chip model, where we have a layer of intestinal organoid cells and then a vascular layer that is composed of endothelial cells and macrophages. And we want to study the interaction between these two layers and also see if the macrophages are recruited to the site of infection or not. And for this, we use dynamic 42 biochips that allow this tissue-to-tissue interaction between the two layers. So electroporation of the organoids was the most efficient method to replicate the virus in our model. As you can see here, we have an increase in the viral load over time. We also then infected naive 3D organoids with the virus that were in growth media, which means they are more proliferative status, or in differentiation media. And we also see that the virus replicates to higher titers when the organoids are in this proliferative state. And thinking on that, we wanted to see what is the tropism of the virus. So we did stainings and we found out that the virus co-localizes with proliferative cells. So these Ki67 positive cells. And when we quantify from all the infected cells which cell type it was, we could see that in infected conditions, over time we have an increase on the percentage of proliferative cells that are uninfected. And at the peak of replication, almost 2/3 of all the infected cells are proliferative. So this hinted that the virus really relies on this process of cell proliferation to also replicate and grow. And also based on this, we wanted to see, are the particles infectious and also which type of virion, because HEV exists in two types of virions within the host, a naked and a quasi-enveloped form. And when we did a gradient centrifugation to look at the type of virion, we see that the virus that is produced in the enteroids has a density typical of a naked virion. So if we think at the way that the virus is transmitted, this hints that the gut is actively producing naked virions that are secreted in the feces, that will then be responsible for the transmission of the disease, but also that could reinfect other regions of the gut or cross the blood barrier and go to the liver and actively infect the liver over and over again. When we moved to the gut-on-chip system, and we saw that the virus is secreted more apically than basolaterally. And here we see a decrease in the virus over time because we exchange the media every day completely, but we still see that every day the virus keeps being secreted into the supernatant. When we convert these into the cumulative, which means the total amount of virus that is secreted, we see that as an increase in the viral load over time. And this bigger secretion into the apical site is again reinforcing this hypothesis that the gut keeps feeding virions into the feces that will then be secreted, and that could also cross the barrier and go to the liver. So we are now also working on establishing this multi-layered gut-on-chip, that also we have this endothelial layer, because now we want to look at the dissemination of the virus and see if the virus, when it crosses the blood barrier, does it also infect endothelial cells or not? And also, are the macrophages infected? Do they interfere in the answer to the infection? Are they recruited or not? So for that, we will build on this gut-on-chip model. We'll also build a liver-on-chip model, and we will connect the two compartments to see the full dissemination of the virus in the gut-liver axis. If you are now starting with the organ-on-chip project, I really advise the use of human-derived cells, like human organoids or human primary cultures, so that you can really take advantage of using the complexity of these cultures and the dynamic advantages of having this microfluidic system. In our case, for example, we could see just the impact of this shear stress on the gut layer when we compared a chip that was in static conditions versus one that was in perfusion. In static, you really see a typical monolayer with all the tight junctions and close epithelium, but a very thin layer. When we compare with the perfusion layer, we see these villa-like structures protruding out of the layer, and we also see the thickness of the layer. So this is just the impact of the shear stress on our cell layer. And if you use physiological relevant cells, you can take even more information from that, because also this impact of this shear stress, the molecular gradients, we also saw that they simulate the differentiation in mature cell types, and you can really have this complex system that are now, we can really mimic in vitro what happens in our gut. And I think this is a very big advantage, not only for gut, but for other culture models that you would be using. So my advice would really be to use human-derived cells, non-immortalized, to really take full advantage of using such a complex system.